Hi there. Today we will be talking about the Varicella zoster virus, which is also known as chickenpox. This is Danielle Nelson for Microbiology Bio 204 with Professor Jaimie Robonson-Bergstralh. See if I can zoom in here for you. So we start with the Varicella zoster virus. It's exclusive to humans, so it's a little bit difficult to study in animals. However, they have been able to graft skin onto mice, human skin onto mice, to study its mutations a little bit better. First off, it's a very infectious virus. It's transmitted through airborne respiratory droplets, like a cough or a sneeze, or if you come into contact with the skin blisters or lesions. Not sure why this picture is not showing up there. Symptoms arrive around 10 to 20 days after exposure and usually present with a rash, fever, typical illness type symptoms. Once you've been infected, about 14 days later, 10 to 14 days, you should heal, and then the virus becomes latent, which means it still remains in your body, but it's not active. And this particular virus sits in the cranial nerves, the dorsal root ganglia, and a lot of different nervous ganglia, I guess you could say. Upon reactivation, this really only happens in elderly people or people that are immunocompromised. It's not quite sure why that happens, but when it does reactivate, it can become different forms. The most common form is shingles, which is like a blistery rash on the skin. It's very painful because it's kind of attached to nerve endings and shows kind of a burning effect. I had a picture here for that. I'm not sure why it's not showing up here, but I'll attach it with my initial submission. Several other things can happen towards the reactivation, anything from inflammation of the eye and vision loss, blood vessel abnormalities. Really seems like there's a lot of things that can go really wrong, which I was unaware of just from being exposed to the virus. Let's see here, mutation, we have glycoprotein gene mutations. That's kind of all I was able to find. Upon infection, a strong immune response is elicited by the body. Here, this is just a picture of the glycoprotein E, which is the one that is responsible for triggering that immune response. Excuse me. Over here we have replication. Once it enters the body, it mostly replicates in the tonsil area, spreading to the tonsils and lymph tissues, and then it accesses T cells in the body, which is where it will replicate and travel throughout the body. This here is kind of a very scientific way of explaining how they replicate. Membrane glycoproteins have large terminal regions, and this enzyme in the VZV contributes to the replication. It's presumed that glycoproteins bind to the cell surface receptors during the attachment. Then the GB complex, which is the glycoprotein B complex, fuses to the virion membrane, which is the virus membrane, and endocytotic vesicles or directly with the plasma membrane. So it's basically kind of just invading that cell. Then once it's invaded, that capsid is released into the cytoplasm and then initiates gene replication inside that cell. Let's see, nutrition, as far as that goes, I was able to find it really just uses the T cell, the mechanisms inside the T cell to replicate. I didn't find a lot of nutrition specifically for this virus, but it does reproduce, or it's synthesized in the endoplasmic reticulum. And then those glycoproteins can undergo further post-translational modifications, such as phosphorylation, ubiquitization, and that might also function in the varicella virus replication. This is an image of glycoproteins being, quote, trafficked. Can't really zoom in anymore there. out of the cell where they're made, and then they are exocyted out of the cell. I hope that gives you kind of a good site map of this virus, and I hope you enjoyed. Thank you.